these sequencing data sets and all the analysis that we have done so far across different functions at Obsidian actually allowed us to understand our lead product, which is the OBX115, at a really great depth. And I do think that this is a huge win. Having a therapeutic in hand and understanding every single side of it is not easy. But TCR sequencing is actually helping us do that.
Hi, everyone. A couple of years ago, I got completely obsessed with T-cell repertoires, and I know I'm not the only one. And that's why I started this podcast. Welcome to the Repertoire Room. My name is Sander, and I'm the co-founder and CEO at ImmuneWatch, an immunoinformatics company specialized in T-cell receptor sequencing analysis. And in this show, I want to have insightful conversations with people who share this passion and go deep on T-cell sequencing technologies and analysis. And today on the show, we have Arman Aksoy, Director of Computational Biology at Obsidian Therapeutics. Welcome, Arman.
Thank you so much, Sandra, for having me. Thank you.
Yes, I'm super excited to have you here, but let's first introduce you to everybody else here. What is actually your current role?
Yeah, thank you. So as you mentioned, I am the Director of Computational Biology at Obsidian Therapeutics. And my role is actually coordinating data generation and analysis efforts for early discovery, preclinical, and translational and clinical functions. As you can imagine, as a startup, we are a very lean team. And actually, my department consists of just one person, which is myself. And I'm a very hands-on computational scientist who is working with at least 10 more others at the company that are all computational savvy people. And then they do all contribute to the computational biology work.
I like the one person department description. That's really fitting for a startup culture, I would say as well.
Yeah, I think times are changing as well. We have this new wave of scientists who are coming, into companies and academics with the computational knowledge to them. So I think the computational biology scenery is changing all the time.
Yeah. Well, that's actually also a good segue to my next question, because I myself have a background in bioinformatics, and I also think there's multiple paths into a role like computational biology or bioinformatics. So I'm also very curious, what's your origin story? How did you actually end up here at the interface of T-cell therapeutics and computational biology?
Yeah, great question. And unfortunately, it's not a straight path. It wasn't a straight path for me to come into the computational biology and at the intersection of immuno-oncology. I'm actually originally from Turkey. That's where I lived and studied until the end of college. And back in those days, believe it or not, we didn't have formal bioinformatics or computational biology departments or training programs. But I was really interested in the computational side of things. So I ended up actually double majoring in cell biology and mathematics, which was, not a standard thing to do. The advantage of this was actually me becoming a hybrid scientist, because as part of my college years, I worked in a wet lab. And then the focus of that was actually neurodegenerative diseases, which was a lot of great experience for me. After that, I got accepted into a graduate program at the Memorial Sloan Kettering Cancer Center in New York City. And I always consider myself as a really lucky graduate student, because I academically grew with the Cancer Genome Atlas project, which is a massive genomics project with the cancer patients. And my thesis lab was actually heavily involved with the projects. And I essentially became a very hardcore genomics person with a very deep understanding of how do physicians take that patient omic data and then inform therapy decisions in the clinics. As I mentioned, I was lucky because this was all happening at the Sloan Kettering. And to be honest, up until this point, immunology was a field that I was very scared of. I thought that it was super complex, and I wasn't actually imagining myself going into immunology at any point. But during my postdoctoral work at Icahn School of Medicine, actually, another amazing thing happened, which was the rise of the checkpoint blockade therapies, all the anti-PD-1, anti-CTLA-4, and therefore I found myself in the middle of immuno-oncology world just by chance. And my specific focus was actually on the biomarker discovery for landmark checkpoint blockade trials, because there was a lot of trials that were happening at that moment. And our lab was focusing on personalized vaccine treatment, immunology-related tool development, so on and so forth. So I essentially, found myself in the immuno-oncology world, and our lab was a computational one, but it eventually turned into a hybrid one because we always wanted to follow up on the computational hypothesis or the things that were coming out of our analysis. But as you can imagine, it's hard to find collaborators who will do just experiments. So we got lucky, and then we had an extension, wet lab extension to our lab. And because of my wet lab background, I ended up going into the lab again and just focusing on T-cell biology, specifically adaptive T-cell therapies. And I know that you think about TCRs a lot, and one thing that I'm really proud of from that area is actually discovering 4 novel murine TCRs that were all reactive against MC38 cell line. And that was all thanks to just understand the computational side of things and then trying to focus on a simple question. And after this, I was in the immuno-oncology world. I first joined Agenus, where I was a computational biologist responsible for scaling and optimizing the high-throughput T-cell profiling platform for them. I then joined Arman Therapeutics, where I was the founding computational biologist, and I was co-leading the target discovery efforts. And I eventually ended up at Obsidian, and that's where I still work.
What a journey. It's actually not the first time that I hear about like a pure computational lab, then also venturing into a wet lab because of necessity. And I think having both skills, as you mentioned, like a hybrid scientist, it can also sometimes work in your advantage on the data analysis side, because you might be able to figure out where things in an experiment went wrong. Is that something you experienced at that time?
Yeah, I actually wasn't thinking that this was going to be a super advantage, because sometimes people think that the wet lab work is just the time sink, but actually it's not. You hit the nail on the head, which is if you see how the data is getting generated, if you're familiar with all the protocols, all the sample preparations, all the things that the wet lab scientists are struggling with, actually it makes the data generation much more efficient. And actually it also helps you with the data analysis, because you understand the sources of error. That can be simple as like a sample mix-up, or that can be as simple as like a poor extraction of the, for example, DNA or RNA material. So yeah, I did hybrid work, which was not that easy, but it's actually helped me a lot becoming a better data scientist or computational biologist.
Yes, I can really, really relate to that. Yeah, interesting. Okay. Now you mentioned also you joined Obsidian Therapeutics. You were not there from the start, but I'm always a little bit interested in like the origin story. So how did Obsidian Therapeutics actually get started, and what problem was it initially trying to solve?
Yeah, really good question. And as you also know, like startups change their profile time to time, depending on the needs, depending on how the research goes. So I might not be the best person to talk about the beginnings of Obsidian Therapeutics, but I should note that there's a really great The Long Run episode where Luke Timmerman interviews with Tariq Qasim, and then he talks about the fun story behind the company. Actually, when I joined the company, Obsidian was already 7 years old, and it was a Series B company that was still at a preclinical stage. As common to most pre-IND companies, the data generation efforts were at their peak. And our CSO back then, Yantar Milan, was a huge believer in making computational biology one of the pillars of the research and development, because he knew that we needed to establish a data-driven organization who is trying to nominate and advance the lead program into the clinics. So that's exactly the time when I joined Obsidian, where data generation and pushing the programs into the clinics was a priority.
Okay, that means that you got some exciting work. You were able to start off some exciting new work within the company, I feel. Okay, and for those less familiar, what does Obsidian Therapeutics then do today? Because there were some pivots, it's data-driven now, but what is like the core activity at Obsidian?
Yeah, so at Obsidian, we consider ourselves as a cell and gene therapy company. And then, of course, we are here to deliver transformative outcomes for patients. Our current lead program is OBX-115, which is an investigational tumor-infiltrating lymphocyte cell therapy. I will try to use TIL, shorten that to TIL therapy as we continue talking about this. But one thing that's unique to our lead program is that we modify these TILs and then put a membrane-bound cytokine, which is IL-15 actually, into them. And then that actually helps us a lot, because that doesn't require additional support of IL-2 for the patients, which is pretty common for conventional non-engineered T cell therapies. And also that removes the toxicities that comes from the IL-2 usage. So we also have another component where we actually have ability to regulate the membrane-bound IL-15. So with an additional drug, we can actually fine-tune how much IL-15 these cells have. So right now, this lead product is in the clinics. We are in the phase 2, and as you can imagine, we think a lot about our current lead program, which is OBX-115.
Yes, I can definitely imagine going clinic phase 2. It's interesting times then for the company.
Yeah.
All right. One of the reasons I wanted to talk with you is, of course, I also know this is a podcast about repertoires, and I also know that you do quite some repertoire work at Obsidian. So I was curious whether we could have the conversation indeed about how, for example, T-cell repertoires are being used at Obsidian.
Yeah, thank you so much for asking. And as you can imagine, as a TIL therapy, we think a lot about the tumor-reactive T cells and the polyclonal TCR repertoire that they make up. And I think one nice thing about this whole field, which is TIL therapy, is that we take the tumor chunk and grow the TILs out of it in multiple stages, and expand those cells to billions of cells, and we use that as a final infusion product. That actually gives us a chance to profile them very deeply, and then, as you can imagine, that final product is a polyclonal T cell population with a lot of TCRs in it. And another interesting thing that happens with the TIL therapy is that most patients go under lymphodepletion before they receive the TIL therapy. So these infused TILs will have a chance to remodel the TCR repertoire in the systemic circulation and in the tumors until the immune system reconstitutes and establishes a new equilibrium. And then that's great, because that gives us a chance to actually change the environment so that whatever is remaining would hopefully be tumor-reactive clonotypes that are capable of, mediating a durable anti-tumor response. As I mentioned, the infusion products gives us a chance to actually do, for example, TCR sequencing, and we do this all the time. And then, because we can understand what is in the infusion product from the TCR perspective, we can actually keep track of those individual clonotypes in the circulation and in the tissues using samples that we collect from the patients during the treatment. This is a really amazing data set, because we can then ask questions like, are there clonotypes that were in the infusion product that are getting enriched or depleted after the infusion? What do they do after they are infused into the patients? How long do these T cells persist? How long we can detect them in the circulation or in the tissues? And also, we are, of course, interested in all the TCR-related metrics that can help us define a biomarker for responsive therapy, which is a pretty common thing for most of the TIL companies. And again, as you can imagine, this type of profiling is one of the key components of our translational efforts. I know that we won't have enough time to go into details, but we have recently presented some TCR sequencing results at ASCO and ESMO this year. So I highly encourage those of you who are curious about these initial findings to go check out our website for a copy of the presentation materials.
It seems like a lot of interesting data to be able handling with a one person department. It seems like very interesting research questions, especially like indeed wiping out the whole immune system and then seeing if you actually introduce some of these TILs again, how the immune system handles with this. I think this is probably interesting data for you to look at.
It is definitely, yeah. And also, I think it's really important to understand different functions, goals. The clinical side has a different goal, translational side has a different goal, preclinical discovery work has a different role. And without understanding how the data will be used eventually, it's really hard to just generate the data and then let it sit. So this has been a really major learning experience for me, as well as I transition from the preclinical work to clinical work. I now appreciate how the data is getting used and how that can play an important role in the treatments as well.
Yeah, and that really helps, I believe, in your experimental design, because you have research questions that you want to answer, you know what you might be expecting. So I think you can better design now that you know what you're looking for, I think you probably have a better idea of what you want to design.
Yeah.
Yeah. That also brings me, maybe in general, when I talk with people in the TIL field, I see a couple of reasons why people would apply TCR sequencing. I think you mentioned some of them already. For example, one of them is more an immunomonitoring strategy, but you also see some people using it as a quality control for the product. Where do T-cell repertoires play the most crucial role at Obsidian for you?
I will say both of them are really important for us, but I should also add process development to the mix as well. Being able to profile T cells within the process also helps us understand what's happening to the TCR repertoire at different stages. So I think, having said that, people consider that TCR sequencing data is core to all of these three efforts, but also as a field, especially within the TIL therapy field, things are changing very rapidly, and the data coming out of different industry and academic efforts is very at the early stages. I think as a field, we are still learning how to best make sense out of all the endless combination metrics that can be derived from the TCR sequencing data sets. And and we also need to understand what each metric might mean for process development, quality control, and translational efforts individually. So as we are learning, I think people are understanding the value of TCR sequencing, and I do think that that's going to stay, in the field as a very powerful tool that we all will use. But I think there's still a lot of learning that we need to go through before, we have standardized places or standardized protocols where we can use it for every single one of these stages.
Yeah. Sounds very reasonable. And also maybe a good follow-up question in that regard is that you now went through all of these hurdles. There's still a lot of learnings. And there's still a lot of things to be discovered, but you also have maybe some learnings. So that brings me maybe to the question on what were kind of the biggest hurdles you had in implementing T-cell sequencing technologies. Maybe this could be interesting learnings for the rest of the community here.
Thank you, and buckle up because this might be a bit of a long answer, but this is
now I'm curious.
Yeah, and this is the heart of computational biology and computational efforts, where there is a specific data set that you need to generate, and there needs to be a lot of coordination and understanding that needs to happen upfront. So, where should I start? I guess, first of all, and again, another high throughput profiling, technique that we're talking about, which is TCR sequencing. So it is really important to understand that you need to plan and run feasibility studies upfront before just taking some samples out of the freezer and then sending them to TCR sequencing. And one thing that people usually don't understand is that this takes a lot of exploration and testing different platforms, different sequencing settings, even shopping around vendors, or maybe trying different computational tools side by side for all of these feasibility and proof of concept studies. It is a lot of work, but skipping all of these is a huge risk, and then that might end up actually your TCR sequencing results being not that interpretable. I think we talked about this, but different questions might require different sequencing settings. It can be different starting material, different sequencing depths, maybe different chains to sequence even. And this is where people need to really sit down and then think about what to do with an internal team if they are doing TCR sequencing in-house, or an external partner that they're using for TCR sequencing. We are lucky. We've been heavily relying on our Repertoire for our TCR sequencing needs, and they've been an amazing partner so far. We have established a really robust and streamlined workflow with them. And thanks to this relationship, we now have a really reliable and standardized internal TCR data set, so that we don't worry about generating data set and that not being compatible with the historical ones. And we do mine that data that we generate all the time for scientific, translational, and operational questions. So that's the first part. That's pretty common to most of the high throughput platforms. But I think another important thing is being really proactive about TCR sequencing. What this means is that people sometimes use TCR sequencing as an afterthought, where they say that, oh, if something goes wrong, if I need to troubleshoot something, I will just take some samples and send them out for TCR sequencing. But I think that's the wrong approach. What I do think needs to happen is you need to think that everything will be going wrong. So you need to prioritize and standardize your sample collection by making it part of the experimental plan. And this is where it becomes important to talk to the people who are going to do all of those procedures and run all of those experiments upfront. And I think this is important also because you need to build a critical mass of internal TCR data so that when the new data comes in, you have a sense of, oh, this looks similar or dissimilar to the ones that I had. And that also takes a lot of efforts and resources. But unfortunately, people, again, skip this. But it is very crucial for all the comparisons and analysis down the line. Third one, I think we also talked about this a bit, but TCR-seq dataset is a very complex dataset. There you can individually focus on clonotypes, or you can take a broader view and get some summary metrics out of it. Like people talk about clonality, diversity, some indices that they use in different places. And there are really great libraries and tools out there where you can take your TCR-seq and just generate all of this. But without knowing which metric to keep track of, the level of noise that's expected and how a real biological difference might be represented in the data, I find it easy to get lost and misinterpret the changes in the metrics. This, again, keeping track of one clonotype is very, very easy. So of course you need to find some sort of summary metrics so that you can just take a look at things across hundreds of samples and then at scale. But doing so requires really extensive understanding of how those individual metrics behave at baseline or when the samples are perturbed. And the last but not least is actually the systematic annotation of TCRs. So it's nice to have these one metric that you use as a summary from all of those clonotypes, but also there's a lot of information that you're not using if you're just focusing on that. So in our case, for example, we care a lot about the clonotypes that are likely to be tumor reactive, specifically reactive against melanoma antigens, for us. But going from a TCR sequence to its putative target is not an easy task, and I'm sure that you will understand this much better than anybody else.
I know about this field a little bit.
And in the past, actually, we used to look up these CDRs that appear in our TCR sequencing data sets using public data sets like VDJ, DB, or so. And sometimes we search them, within other TCR-seq results, like, for example, ImmuneAccess, to get a sense of what each TCR might be targeting and how we can, build information or stories around it. But again, as you can imagine, these exact lookups are not that helpful because, you need to put some heuristic into it so that, you can actually find something relevant to what you're looking for. Again, there are lots of libraries and tools out there that can facilitate this, but especially with just basic things. We have been doing this on our own, like sequence clustering, annotation transfer. We had some implementations on our own, but I should say that this does feel like chasing the red queen. Those things that you use change all the time. Public data sets are constantly growing. There are new resources that are getting released every single day. TCR sequencing solutions are getting better and better. So having a solution that's live and applicable all the time in-house is a hassle. We talked about this, and we are a small team and it's a very early stage biotech company. So it is not an easy task to just tackle all of this with the small group of people. So this is where we found actually some relief, especially after switching over to ImmuneWatch Detect approach. And doing this through our sequencing partner, our Repertoire, helped a lot. instead of talking about all the things that you did to analyze the data, find targets, or maybe nominate targets, it's very easy to say that here's an award-winning algorithm that people rely and trust, and this is what we are using. And then that allows us to not worry about the changing data sets, like, keeping track of everything that might be improving. And that, essentially makes things much more easier for us. And we do understand that this TCR to epitope database is very sparse, and it's going to stay sparse maybe for a while. But until it gets saturated, being able to rely on a solution that will give you the best results is going to be crucial. And that trust that you get from using a solution that's being proven, I think makes a huge difference.
That's what we're hoping to achieve. So it's nice to hear that from you. But what I also remember, or what I take away from your answer, is it really shows that you're a hybrid scientist, right? Because you start your answer with saying that it's really important to get the foundations right. The sequencing methods have to be right for the questions that you want to solve, and then you build up to what computational tools. But the core is really nailing down what specific way of T-cell sequencing do I need to do in order to answer the different research questions that we have within the company. So I feel that you covered everything from like the wet lab part until the dry lab part in this whole answer. These are very interesting takeaways, I think.
Thank you. I hope that this is helpful. I think people do oversee these when they sometimes, for example, switch platforms or TCR sequencing methods in between. But we try to make sure that whatever we use for preclinical is also applicable for the clinical, and so that you can actually combine all of the data sets. And yeah, I think having somebody who understands the experimental and computational sides is really important here.
Yes, I 100% agree. We talked about the lessons you learned. Now, maybe I want to go a little bit more deeper into, you've implemented all of this, you did a lot of effort in implementing T-cell sequencing, did a lot of comparisons as well. Did it actually make a difference for Obsidian in general? And if so, what kind of difference?
Yeah, great question. You generate all of this data set, you spend all the resources and time, but what do you get out of it? So I do ask this question to myself a lot too, because you need to understand that you're generating all of this, you're building a database. But one thing that I can tell for certain is that all of these efforts, all of these sequencing data sets, and all the analysis that we have done so far across different functions at Obsidian actually allowed us to understand our lead product, which is the OBX-115, at a really great depth. And I do think that this is a huge win. Having a therapeutic in hand and understanding every single side of it is not easy. But TCR sequencing is actually helping us do that. Like we talked about this, but immunomonitoring, keeping track of things at the process development side. Also, having systems at the preclinical stage where you use these TCRs to inform what you're going to do is really important. So I think you ideally want to be at a place as a company to generate all this data so that you can troubleshoot things when needed, and you can use this data to improve things. And we talked about this. It takes a lot of coordination, and in our field, people have this joke of like it takes a village to get to where you want to go. And
okay.
So I think I'm proud and lucky to say that this is where Obsidian is right now. Right? We have an amazing TCR sequencing data set that we mine all the time. It helps us troubleshoot things. It helps us improve things. But I should also note that, like, we talked about this a bit, but the field is growing. The field is learning how to make use of the TCR sequencing. But there's also another complication, which is that there are different flavors of TIL therapies out there, right? There are engineered ones, there are unengineered ones, there are enriched ones, not enriched ones. We have different molecules that gets engineered into the cells. And we now know that like TCR metrics that are applicable and meaningful for one might not be actually applicable and meaningful for another. And this is a really important thing because This is why we needed to build our own TCR sequencing database and re-evaluate the meaning of different metrics. So unfortunately, it's not that easy to take one metric and then use it within the same context as others do. I won't be able to go into details of everything due to limited time and scope of this, this discussion. But one specific example that I can point out is, our product is predominantly CD8 positive T cells, and with that comes a huge difference in the diversity and clonality metrics that you have. We are working at a different scale when you are talking about these metrics. And then this is something that's different from what others are seeing and reporting, as part of different studies. Another example that I can mention is, like when we get lucky and have a high confidence prediction, for example, like one that's being, coming out of the DECT with an experimental support, where we see a TCR being predicted to be reactive to a melanoma antigen. That's a huge win for us because it means that we have a clonotype that we can keep track as part of our, for example, process development, preclinical efforts, or within the clinics, where we get an understanding of how the dynamics work out, right? How that individual clonotype behaves in the patient or in preclinical models, for example. And that actually allows us to go back to the data set. And as we discussed, the data set and the predictions are not complete, but knowing how a tumor-reactive clonotype behaves helps you to generalize and go back to the data set and look for those patterns where you try to look for other tumor-reactive clonotypes that are not predicted to be. But you can then group them and then try to get a sense of like how they are behaving, how good they're persisting, so on and so forth. So all of these little things, when you're talking about it, actually makes a huge difference because, as I mentioned, it helps us understand how our product is behaving in preclinical and in the clinical side of things.
You're starting to make me feel jealous. Like I really, I'm starting to want to work at Obsidian to tinker around with these data sets. That sounds like really, really interesting insights, especially like the tracking, finding some maybe highly high scoring annotations and then tracking these. That seems super interesting. Yeah. Okay. It seems like it was worth it already doing all of these efforts.
It is, yeah. I think when I talk to people, and then when people ask me like what is like something exciting for you, is actually just this, right? People waiting, and then looking over your shoulder for some interesting results to emerge from the data sets. And I think sometimes people think that this whole computational work is a black box where you take all of the TCR sequencing data, just point out to the database and get some insights out of it. But actually, it's not. Like as a field that's growing and just like learning, I do think that it takes a lot of effort to find some anchors in your data that can then help you generalize things and then start to build stories around that. But yeah, that's the goal. I think I'm lucky to be at a place where people are doing that, where they just wait for computational, results to emerge, and then we have discussions around them, and then we go back and then iterate.
Yes, and the more experience you build on your unique data sets, the quicker you'll find these insights and these anchors that you can work with, I can imagine.
Yeah.
Yes. You also mentioned a couple of times, indeed, the whole field is evolving, specifically also for the T-cell epitope annotations, like what we do at ImmuneWatch. There's still a lot of, also in general, just an evolving field. It's a tricky question, I think, but in your opinion, how do you see T-cell sequencing and analysis evolving in the coming next years?
Yeah, a really great question. And, to be honest, it's always hard to predict what's going to be happening in the coming years because there are always these like major surprises that completely change the field,
That's true.
For instance, but, but I think, from where we stand, we talked about, the TCR, for example, to epitope, mapping data sets. Like they are very sparse and people know that, people understand that. So but people also understand the value of those annotations. Like I just mentioned to you that even finding one hit helps you understand the data better because then you have something that you can go back to the data with some hypothesis or with some observation or with some pattern. So as people understand the value of this, I think our field will find ways where we can optimize experimental systems and try to saturate the data sets like much better than we do right now. And I know that this is something that you think a lot about ImmuneWatch as well. This is the data set that we use for predictions. We have a tool that's doing really great. It's making us make the best out of the limited data set and expand our scope. But we can do better, right? I'm really looking forward to the new platforms that are cheap and efficient, that can help us generate the data set so that all of these tools, all of these amazing tools will do a better job. But I know that will take some time and efforts, and again, like a lot of resources as well. Until then, I think I'm now seeing an increased interest and some usage of single cell sequencing, where people try to get a sample of the tumor, the primary tumor that the TILs are grown out of, and then they try to take a look at the gene expression or transcriptomic profiles of the T cells to get a sense of if it's tumor reactive or not. And I think that's an interesting new development that's again evolving right now in our field. But I can imagine this can help annotate and predict clonotypes that are likely to be tumor reactive by looking at the tumor itself instead of purely relying on the TCR sequences. I think this is not going to replace like the TCR sequence-based predictions, but I think it's going to be a really nice complementary prediction approach. But one thing that's hard with this approach is that you need to process the starting tumor material for single-cell sequencing, which is not that straightforward. And it's, again, another thing that you need to have really amazing coordination across multiple teams, right? You cannot take one tumor chunk from the freezer and then just go for single-cell sequencing. You have to plan for it upfront. We actually started to explore this approach at Obsidian. We actually had a poster last year that talks about the single-cell sequencing-based tumor reactivity scores. But I think, again, this approach is still in its infancy. But I do think that as people build more data sets, try this approach out, I think we will have better ways of doing things. But eventually, it's not going to be just one thing that will take the field, dominate the field. I think all of these simple things will come together and will give us a really nice integrated solution where we can take the TCR sequencing that we generate and then be able to put some more insights into it, or we can extract more insights out of it, maybe I will say. So in an ideal world, hopefully maybe in a few years, we will be in a place where we take a population of T cells, TCR seq it, and then understand what their targets are and how they're relevant in various diseases and what their therapeutic potential are. Hopefully there will be a place where we will be in a few years.
I agree. I would also sign up for that. My final question, this has been extremely interesting as well. I always have this final question. It seems that you're very passionate with your job, but I would also be wondering what you were doing if you were not working as a computational biologist, Arman.
Yeah, so this is not going to be a surprise, but definitely that work.
Okay.
Yeah, not another job, not another thing, not another place, but actually doing experimental work. I really miss the old days where I was doing hybrid work and switching between experimental and computational tasks during the day. For the last 4 or 5 years, I've been predominantly focusing on computational work, and it's probably for the best because I probably have already lost my edge at the bench. But I hope that, in the future, I'll be at the place where I'll be able to just explore some experimental things on the side and then come up with different computational approaches based on that, just to optimize the data generation efforts. But one can hope, but I don't know what the future will bring.
Okay, so really, truly, again, speaking to the hybrid scientists, you missed the wet lab part of the hybrid scientist hidden inside of you.
Yeah.
Arman, this has been great. I've learned a lot of new things as well. I really appreciate that you took the time to have this conversation. If people want to find out more about you or about Obsidian, where can they go to connect?
I think our company website, which is obsidiantx.com, is the best place to learn more about Obsidian and connect with us. As for myself, I'm pretty active on different social media platforms like Blue Sky, LinkedIn, X, GitHub, Hugging Face. Searching for my name and just like finding me on any of this will be the best way to connect with me. But yeah, thank you so much for having me. This was a really great discussion.
Thank you. I would, by the way, encourage everybody to start following Arman, because I've been following on LinkedIn as well, and he has some very interesting vibe coding experiments that he shares. I'm always very curious when you have these posts. So, except for next to his interesting work at Obsidian, I think this is also worth a follow.
Thank you. That's what kind of you say.
Thank you for being here, and I hope to speak again in the future.
Thank you. Take care. Bye-bye.